Thank you, everybody. I'm really excited to be able to be here. Unfortunately, the results of this trial are embargoed until Monday, so please come and hear the talk for the first time. But I'm going to talk about some of the operational considerations, participant considerations. So my talk is entitled Implementing, well, whatever you read, that was it. For those not familiar with the FRESH cohort, we're based in KwaZulu-Natal, South Africa. Next slide. And our original aim was to enroll young women in a trial and do very, very frequent monitoring. These are women not living with HIV at high risk of acquisition. And to test them so frequently that we're going to pick them up at the earliest possible time point of acute infection. So we can study those early events that are important for developing a cure intervention. And that for some people, you're like, oh, wait, you're just monitoring them and waiting for them to get positive. It's an extremely intensive prevention program, too. There's a lot to it. But that was the original. We wanted to, during the exponential viral load replication phase, detect people and get multiple samples before they ever reach peak viremia. Next. So the program, we enrolled, again, these women not living with HIV in a 9-month program where they get their fingerprint for an HIV RNA, for viral load, twice per week, and it's combined with an empowerment program so that the women have an immediate and tangible and sustained benefit from doing the trial. It goes beyond receiving a cash stipend. Next slide. Okay, so Cindy and I work so closely together. I'm at the clinic, 20 minutes away, Cindy in the laboratory. So we really take the basic science that he's working on and communicate back and forth with his team what we're doing in the clinic and feed back to them also. Next slide. All right, so before we even had a trial, before we even dreamt of being a trial site, we were already focusing on the patient, and this was very early on after starting FRESH, which we started back in 2012. Combining basic science with social good. Next slide. Okay, so there's Subi. He does leave the lab occasionally and come to site and speak to both participants and to the staff. Next slide. And we want investigators, whether they be from the US or from Africa, to interact directly with the trial participants. So before they've even conceived of the new protocol, try to understand the population that we're working with. Next slide. At FRESH, using this approach, we detected 109 hyperacute infections. And again, this is so early. This is 4 days since their last HIV RNA was negative. So this is the big one. The first 14, this is when the guideline in the US was still ART was you're only eligible for ART when your CD4 count dropped to 500. In South Africa, it was still 350. So those first 14 women, we weren't allowed to treat them. We applied for permission to treat them, and we got that permission. And as soon as we did, we started offering immediate treatment. And so we've offered now to 95 participants immediate treatment. All but one has taken that up. And so we now have a cohort of very early treated participants who have a small, non-diverse reservoir and should be very interested in having cure trials. Next slide.
Wait, sorry.
Okay. So we have an observational study, and we've now become a very vigorous trial site for both prevention interventions, not just cure, but also cure trials. Next slide.
Hold on, she's still quick.
Okay. So this is the design of the ATI trial that we just finished on January 15th, last patient, last visit. And you look at these diagrams, it was 20 women. All women are part of the FRESH cohort who started very early treatment and had been suppressed by the protocol for 12 months. But the median time they had been on treatment was almost 7 years. So this is a cohort we've really been following closely and we know very well. The drugs we were using, dual broadly neutralizing antibodies, BRCO7-523LS, CAP256, and a TLR7 agonist, vesitolamod, and that's to kick the reservoir, and this is to trap the virus once you've kicked it out. The blue arrows there are the doses of vesitolamod. The red is the BNAB, And then we go into the ATI. Well, they're still receiving oral vest doses. They finish off the vest, the BNAB washes out, and then they're on a true ATI with no drug on board. When they've finished 44 weeks of ATI, if they remain virally suppressed or have not met ART restart criteria, they can go into an elective ATI extension, which is during period 4. Next slide. All right, next slide. So there are different kinds of ATI designs. There are ones where you start very immediately. Any virus detectable, restart them on ART. That's the safest. But there's a lot we cannot learn from a trial like that, and a lot we can learn with some permissive viremia. And those can be we can mitigate the risk by very careful monitoring. So you can refer to this as a set point ATI, or once you've met a less stringent ART restart criteria. And this is the style of ATI trial that we had at Fresh. Next slide. All right. We didn't know anything. This was our first cure trial, and what did we not know? Well, this was the first in Africa and first in women clade C, so how's it going to go? We had no idea. All right. Also, there's the issue, one of the inclusion criteria was BNAB sensitivity, meaning I take your virus, I take it in the lab and look and see whether or not it's going to be affected by the broadly neutralizing antibodies that we're going to use. Initially, our trial design was you have to be dually sensitive to both BNABs, but we couldn't enroll the trial because we were very surprised to find how many were not dually sensitive. The majority were, it was about 38%. However, there were several that were only monosensitive and some that were not sensitive at all. So how much of a difference is that going to make? We didn't know. There are some trials that some sites that argue against even doing the BNAB sensitivity anyway. There are problems with the assay. Another thing we didn't know is, are women going to want to join this trial? Because there's a lot to these very complex trials, as Michael has alluded to. There's a lot we're asking of the participants. What about disclosure? All right, we haven't yet talked about partner safety measures, preventing transmissions of HIV during ATI. Once you rebound, your sexual partner is at risk. So we haven't talked about disclosure of participant. What should be the requirement of a trial participant? Disclose that their HIV status to start with, but the fact that they're on an ATI. And we can make our own decisions, but we took that to the community under the guidance of Corinne, and we spent actually years communicating with the community. I'll get to the publication of that paper. Next, we had optional lymph node and gut biopsies. In order to get that really detailed, what's happening in the tissues, what's happening in the reservoirs, we need to get these samples. So would participants agree to it? It was optional in this trial. And ATI harms. One of the problems that individuals have is we don't really know the harms related to the ATI in particular. We talk about drug side effects, but what about the ATI itself? What are those harms? What are the long-term harms? And where's the reports that are bringing those ATI trials together? And is it only the participant or only the participant and their partners? Or are more individuals impacted by our trials as well? And we actually found out that there are more individuals that are impacted in a surprising way. And then we learned that you can conduct a trial when there's acts of God, like floods that shut down your site, you run out of power, you run out of water, there's civil unrest, your entire clinic is decimated, and then there's a pandemic in the middle of it. So you can carry on. Next slide. Okay, so why was this particular trial really important to me and myself and to others working with our team? Well, most trials, as Sylvia said, have been conducted in the global north and clade B, in resource abundant settings, different populations, different considerations. Can we do this in low and middle income countries? Women only represent 13% of those enrolled in HIV cure trials. So that was, again, important to us. And again, we ask so much from participants, and there's so much that we don't ask them. We conduct trial after trial without engaging with them. Maybe we engage with activists or persons living with HIV, but somehow they don't seem to change the protocols. So there's a lot we don't know that we need to know. Next. All right, This is back to the women issue, that they're underrepresented, so we were really excited about having the opportunity to enroll women because there are sex differences, gender differences, hormonal differences that impact trials. And so we wanted to prioritize those who are at extremely high risk, and these are adolescent girls and young women in sub-Saharan Africa. Next slide. Okay, so we get these protocols. This is the trial we did. It looked like this. We dosed this frequently. In 2 weeks we did this. But these protocols are incredibly intense. Next slide. This team has to, when you get the first dose of this, there's a 24-hour observation. We don't have a 24-hour site, and it's not safe where we operate. So we have to transport to a hospital in town. Our staff member has to sleep overnight. Wait, we can't make them work 24-hour shifts, so they have to relieve each other in the middle of the shift. The participants have to leave their home, come to site very early, get transported to the hospital, and have these frequent all-night vital signs and clinical checks to make sure they're okay. So this is nothing. When they get the BNAB infusion, it's 8 hours on site. So we have staff that are enrolled in shifts to watch the participant long into the night, and there's a safety issue. How do we get them home? So it's a big deal. Next slide. All right. It was optional, but we're asking participants, we really need to know about the reservoir. So will you let us cut down the bikini line and take out a lymph node? So we're not, some sites do just a fine needle biopsy, which is not very invasive, but if you're cutting someone open in the groin and taking out a whole lymph node because you really want to know in situ, where is the HIV? Is it intact? Is it not intact? Is it replicable? That's a lot to ask. If you're a woman and have a higher BMI, you're at higher risk for having a post-incision infection. So there are considerations. And again, they have to go to this other hospital, have a surgeon, it's a place they're not used to. Our team isn't there. Oh, our team has to be there because they're worried about going there. It's a big deal. So we also get gut biopsies. That's kind of like a colonoscopy, but a little bit less invasive. And we do 30 snips. 30 snips. They're not painful. But again, it's a lot. Wait, we didn't ask them to do it once. We asked them to do it again, before ATI, after ATI, when they rebound. We're not asking them to do it once. We're asking them to do it serially. It's a lot to ask. Next. All right, leukapheresis. I think Mike mentioned this too. Apheresis, you ask someone to go and have these large bore needles put in their arm. You have to hold your arm dead straight for the entire duration. At our site, it takes between 3 and 4 hours, just like this. That gets stiff. But look at this. How cool is this? So from the science standpoint, we have a median of 9.6 billion CD4 cells to study and do these really cool assays. So these are the trial participants, 1, 2, 3, before ATI, after ATI, 1, 2, 3. Some of them are very early at the time of their first detection when they were vitamin it. These incredible samples. Next slide. And to get those incredible samples for the scientists, this is what we have to do to a participant.
2 arms.
It's 2 arms, one that's
In and out. Jesus on the cross.
So we give them access to a laptop to watch a movie, but still, you know, it's a lot. Bedpan, you're far away from home, you're doing this, so you have to arrive early, transportation to the hospital, 4 arms with your arms straight. There might be access failure, which is, again, one of those things like, oh, I had access failure, and then you feel bad because you failed your team. You know what I mean? So there's, it sounds like a little thing, but these are so intense, and there's so many emotions that are involved in participating in those trials that you want to do your best to participate. So when you have an access failure or low flow and you can't get a leukal pack, it doesn't go to the lab, it's another one of these opportunities for disappointment for participants. This is a big deal. These samples and these studies have a human impact. Next slide. All right, so there's Corinne and Dr. Madan Tembu. What's your first year coming with the team? 2019. So she came 3 years before the trial started to start talking to the community and developing the language that's not going to be offensive, because there's so many things you can't say. That all takes time. And incorporate social and behavioral research into a protocol. You need the investigators, we need the investigators to engage the social scientists. because otherwise we can't get a protocol up and running in time. The trial started, it's too late. So to get those key time points, the investigators need to talk to the investigators with the social scientists to make sure they're incorporated. So when do we need that? When do we need a Corinne? We need it when we're in a new site or with a new population asking new questions that we do not know the answers to. If you want to help 100 fill in the blank, if you want to help a fill in the blank, ask 100 fill in the blanks what they want. Okay, that takes time. Next slide. Okay, so this was a call to action just to use social science to understand and listen to participants. Next slide. So we want to be patient-centered in these trials, and how do we do that? Next slide. So social and behavioral research has so many ways and so many questions that we, as the clinicians or basic scientists, don't have time or the focus or bandwidth to be asking. Next slide. All right, so back to the protocol. Here's the protocol. So we divided up the many people that we anticipated could be impacted by this ATI trial, and it did not stop with participants. But for the participants at key time points, so before they ever went on ATI, before they received the study product, before they stopped their ART, once they rebounded and restarted their ARVs, and at the end of the study, how did you feel? What was that like? But not just asking them these open-ended questions during an in-depth interview, but asking them a range of questions and finding about their mental health, their levels of depression, their levels of self-esteem, and how were those impacted along the trial? And were they differently impacted if you rebounded early or if you didn't rebound? What were the impacts? Next slide. So here's all the different groups that were involved, from the trial participants, which we focused heavily on, to a control group, if you will. These are acutely treated participants who couldn't join the trial because they didn't meet the criteria. Maybe they were not sensitive to either VLAB or fill in the blank for the reasons they couldn't join. The community, what did the community think? Partners and family members, the research staff. Imagine, if you will, you're in the clinic and you have very strong biases about something that's happening in the trial. That's going to directly impact the way you communicate with trial participants and the kind of support that you're going to be able to give them. So it's critical that we engage them very early and make sure they have a strong understanding and that we understand their feelings before the trial starts. Okay, so this is already been published. This is group 2. This is the control group. The conclusion was that it's very important for us to understand women's perspectives when we're launching into and enrolling more women in trials, and particularly in this setting. Next slide. This is group 3, the community members. This one is also published. The title is "With the Study, We Have Hope That Something is Coming." This is something we learned. They initially, at the beginning of the focus group discussion, they said, it was a lot of men in one of the groups, and they said, you have to make sure the participants are disclosed, you have to protect the partners, very strongly that should be an exclusion criteria. As the discussion wore on, they actually came around and we discussed gender-based violence in the setting. They said, actually, I'd rather that my sister, wife, niece, mom were more protected, and I want autonomy for the participants, that they decide when and if they disclose their ATI participation and their HIV status. So that was something that evolved in the midst of a focus group discussion for them to say they should be the ones that decide. Whereas there's still a lot of PIs who say, absolutely, you have to have double barrier protection, you have to do this, they have to have disclosed. But first off, people, it's difficult to confirm. And also, this is not in our setting what the community felt was the right thing to do. And then back to always simple language and explain it in ways that we can understand. Viral rebound, it's, we see them very fast. They're coming for a clinic visit. How are you doing? I'm doing fine. You feel sick today. We fill out our CRFs. But having that time to sit down and see how are you doing at home is really, really intensive. It can't be something that the clinical study does unless it's part of the protocol. It really isn't, because remember, the trialists that are trying to implement a protocol, that's their job. They're not primary care providers. And there's so many complicated things, like for partner protection, well, the site ought to provide access to PrEP where PrEP access is limited. But this is a non-trial participant. It's not a clinic, primary care clinic, and now we're supposed to dish out drugs that do have side effects potentially to non-trial participants. It's complicated. There are a lot of considerations. So there's disappointment, there's confusion. You told us always to adhere, now you're telling us to stop drugs. So the whole issue of viral rebound and ART restart is a complicated one for participants who are asked to do it. Next slide. All right, so we love our participants. This is the ATI consensus workshop in Nairobi, and this is 10005. Who gave me permission overnight to say, yes, you can call me Charity, in this meeting. And she attended the meeting, and she gave a talk. For those of you who are there, she was extremely nervous, but we were really proud of her. And Michael, thank you for the support that you gave her. But she was there at the event. Next slide. And she was asked on stage, how does it feel to stop ARVs during an ATI? And it's so, I can report it to you, but nothing's better than hearing from a participant themselves. Okay, all right? Okay, next slide. All right, so I need to wrap up. I'm going to be on time here. Take-home points is we should be conducting research where it's needed most. And that does not exclude San Francisco, but it does include Sub-Saharan Africa. And it does include all of those of us who are impacted strongly, and those of us who need and want a cure, who should be involved in these studies, have the opportunity. Basic science and clinical research is incredibly strengthened by working together hand in hand with social scientists, so we can address critical challenges, whether it be poverty, unemployment, food insecurity, rape, gender-based violence, teen pregnancy, whatever the issues happen to be. Build in ways for investigators to engage with participants in the clinical team wherever possible, because that divide between the PIs and participants should be a little narrower, even if it's punctuated and intermittent. And finally, build capacity where there's less capacity. Instead of always reverting to the same sites that have abundant staff expertise, let's build it so we can conduct these trials in other locations. Next slide. All right, so here's two silos, and I'd love to see those merge together, but it's not just product-centered design that we're worried about and the impact of a drug. Does it work, does it not, what are the side effects? But thinking about the humans that are going to give us those answers. Next slide. And Michael, you're controlling this. Can you press play on this slide?
Hello, Karim. I have I'm day 376. I'm so excited.
376 of what? What's happening?
Of undetectable.
And are you taking ARVs?
No.
You're on an ATI?
Yes.
And I'll help
you.
An interruption. Woo!
Hi, Katie. How are you doing? What day are you? I think it's in something. 800 and something. Yeah. I've lost numbers, I think. So 800 is more than 2. Yes, more than 2. Yes. Sing together. Analytical treatment interruption. Analytical treatment interruption.
That was Charity. She's on day 857 today, not taking her ARVs. She says, I forget that I'm sick many times. Next slide.